morning. This video is going to look at the structure of DNA, the different parts that make up the individual units of DNA called a nucleotide, and how the two strands of DNA end up being connected to each other. DNA is basically a long chain of individual units called nucleotides. Nucleotides have three parts to them. There's a phosphate group, which is going to have a phosphate in it at some place. It's more chemically complex than that, but for your purposes, you just need to know that there's a phosphate group. That is attached to one end of a sugar molecule, and that sugar molecule is called deoxyribose. There's actually 5 carbons in the sugar. And then on the other end is going to be a nitrogen base. And there are 4 possible nitrogen bases. It could be an adenine, which we write as an A. It can be a thymine, which we write as a T. It can be a cytosine, which we write as a C, or it can be a guanine, which we write as a G. These nitrogen bases are going to be really what we focus our time and energy on when we're looking at DNA, because these things here are going to give us the information that we need. That is our genetic code. And the relationship between nitrogen bases is what is going to actually hold the 2 different strands of DNA together. So I kind of wanted to show you how this looks if you're looking at a spiraled up DNA here, okay? So the backbone, or the strands of the DNA, are going to be these sugars and these phosphates. They're going to branch out towards the end. So if you think about a ladder, I think DNA is always easy to compare to a ladder. The sugars and the phosphates, they would make up, you know, the ends of the ladder. Okay, so you can see how they branch apart. The interesting part, the part that we're going to focus on then, is the nitrogen base, because they are going to join in the middle. So they would almost be like making the rungs of the ladder. All right, now there's a very specific way that these nitrogen bases combine together. And that is called Chargaff's rule. Erwin Chargaff is the scientist that was responsible for finding this relationship. And what this relationship says is that if there is an A on one of the nucleotides, so let's say there's an A right here, the other side has to be a T. Okay? A's and T's will bond together. C's and G's will bond together. So if there was a G here, there would be a C here. Okay? If this nitrogen base has an A, then the opposite nucleotide, the nitrogen base, has to be a T. Okay? C's and T's, no good. Only A's and T's will bind together. Okay? So they form a chemical bond that kind of holds this molecule together in its wound up sort of shape, and C's and G's. So here's a picture showing you a DNA molecule that's been unwound up. So we've kind of made it more linear, right? So let's say that we have base pairs on this strand. So A, C, G, T, A. Okay? Then we know that this strand of DNA, that the nucleotide's nitrogen base will be the matching pair. So if this is an A right here, this will be a T. If this is a T, this has to be an A. If this is a G, this is a C. If this is a C, this is a G. And if this is an A, this has to be a T. So hopefully you see how we've got 2 strands, DNA double-stranded, okay? There's 2 strands. It is all wound up in a spiraled shape, which is where the helix comes in, is the helix shape of it. And the reason why those 2 strands are held together is because of the bonding relationship between these nitrogen bases. And the phosphate group and the sugars, those would all just make up this black line here, which is, you know, kind of the backbone of the strand. We're not super concerned about the sugar and the phosphate group because it's the same in every single nucleotide. It's the same in this nucleotide, in this nucleotide, in this nucleotide. What we're focusing on is what's happening with the nitrogen base position. Okay, adenine, thymine, guanine, cytosine. A's and T's always join together. C's and G's always join together.